I'm Joe Felsenstein. I want to give an introduction to quantitative genetics in general, variance components, and heritability more specifically. Let me see. I'm also recording, I should say. I'm doing an audio recording of every talk, and we'll post those on the website in two formats. And we're being live streamed, and ultimately those live streams will be converted and edited into videos that are posted on YouTube. And so we will have, here comes that thing again. How do I get rid of this? I mean, this box keeps popping up in front of my projection, and I closed it, and then the zombie, or the werewolf or something, it rose again. I don't have that turned on yet. It'll be back. Oh, I see. Oh, that's right. Because you're right. That's the correct thing to do. I'll turn off my Wi-Fi. And then, okay. So much for. There we go. I think we're fine. Okay. So I want to do an introduction to the general theory of quantitative genetics, classical theory of quantitative genetics, that includes things like variance components and heritability, tell you a little bit about the history. I realize many of you have studied this and know this material very well. Maybe there'll be a few details in it that are new to you. Maybe there'll be a few details in it that you consider to be wrong. And I hope you'll tell me about it if I do. But the other is some of you will, in fact, have not quite enough background, and maybe this will help. And then we'll be proceeding on to Steve's talk, and we will have time for a discussion after my talk, although we have eaten up most of the time, but we're just going to go ahead and do it. We're falling a little behind the schedule, but we'll find ways of recovering. Okay, so let me start out with the classical theory of quantitative genetics and start with historical figures, and particularly start in the era after Charles Darwin, before the rediscovery of Mendel's work in the year 1900. There were people trying to work on quantitative theories of inheritance in natural populations and human populations as well. And predominant among them were Francis Galton, who was a younger cousin of Charles Darwin's, and following him and allied with him, Carl Pearson. And they put forward a statistical theory of inheritance that was basically a bunch of regression equations, regressing offspring on the values of offspring, of a trait in offspring, on the values of the trait in parents. And their formulas almost work. However, they implicitly assumed that what we would now call the heritabilities of all traits were equal, that there was some sort of a universal constant of maybe a half or a third that was the heritability. And so their theory doesn't quite work for that reason. They of course didn't know about Mendelian genetics, and so they assumed that their regression equations were really expressions of how inheritance worked. They notoriously founded the field, the modern field of eugenics. Galton coined the term eugenics, and we know of the sad history of eugenics in the 20th century leading on to the Nazis, which I don't think these guys should be particularly blamed for. They were manic hereditarians who believed that everything, in effect, that everything was genetic. And they did some things that by our standards are now very comical. For example, in order to measure traits in humans, such as the skull shape, they maintained a storefront in London, and they would lurk inside this storefront, and anyone passing their store, they would come out and talk them into coming in and getting their head measured. They accumulated vast amounts of this data. the method of sampling they were using would be regarded as laughable today. But people in London, at University College London years ago, told me that they had like a storeroom full of the notebooks of Galton and Pearson, all these people whose heads they measured in London. The information is now in University College London library somewhere. In the process, in addition to kicking off eugenics, they also did a lot of very important work in multivariate statistics. Galton coined the term regression, and all the work on sort of regression, correlation, multiple correlation, partial regression, and so on, a lot of it comes from their work. A lot of it was pioneered by them. However, then we get to the year 1900, Mendel's work is rediscovered, and in the 1910s, R.A. Fisher sets out to basically redo the Galton's law of ancestral heredity, which was his regression equation, to redo it in Mendelian terms, and basically to say, if we have Mendelian traits, what would the actual relationships be? I think most of you have heard of R.A. Fisher, and he was, of course, a great founder, one of the two greatest of the three founders of theoretical population genetics. In addition, he was probably the most important mathematical statistician ever, and he's responsible for about half of the concepts in mathematical statistics: consistency, sufficiency, consistency, efficiency, a whole bunch of others that I'm blanking on owing to jet lag. And so R.A. Fisher was both a incredibly important mathematical statistician and a very important theoretical population geneticist. In 1918, he published in the Transactions of the Royal Society of Edinburgh a giant paper on what the relationship between relatives would be under Mendelian inheritance. It's a paper which is tremendously important, A, and tremendously unreadable, B. Seminars go through that paper, and it can take them a day to do one page. People have published explanations of the paper and still had people not understand it. It's very important, but it's not light reading. I want to mention one other guy that doesn't get as much publicity, and that's J. Lush. He was an animal breeder. He ended up at Iowa State University. He had contact with Sewell Wright, the other founder, the other of the two greatest founders of theoretical population genetics, and learned the quantitative genetics of Fisher and Wright. And in the 1930s, he went out and spread it to animal breeding, and then it also came to be in plant breeding. And at Iowa State University became the most important center for quantitative genetics applied to plant and animal breeding. That generation of quantitative geneticists had to go out and do battle with show ring breeders who were dominant in animal and plant breeding in those years, in which you brought a cow forward and said, "Isn't it beautiful? We'll give it first prize," instead of measuring the performance of the animal. And there was a whole political battle there that I don't know much about, but someday someone will write an interesting history of it. Okay. So what I want to do is to present to you the Fisherian model, which will be the basis of talking about variance components. And what we're going to do is to assume that a trait is a sum of a series of contributions, and we'll model the value of the trait P as being a starting value, which may or may not be the population mean, but is just some value on the scale. And then the A locus will contribute an amount depending upon its genotype. So here are the three different possible genotypes if we have two alleles, and it will contribute either -2 or 0 or 3. And similarly for the B locus, the C locus, the D locus, and the E locus. So I have 5 loci here. The theory applies to any number of loci. But the assumption made in this simple-minded model is that these terms are additive, that, for example, there are no interactions. We're ignoring epistasis, interactions between loci. And finally, after one adds up contributions from each of the genes, one adds on an environmental effect. And in the simple-minded model, the model basically that Fisher used, the environmental effect is simply drawn from a distribution independently of anything else. It doesn't, I'm not saying that in real life it doesn't depend upon the genotype, but in this case, the environmental effects are being assumed to be independent from individual to individual and independent of all these genetic effects. So it's a very naive model. It allows for dominance, so you can have any pattern of dominance that you want. I've shown 2 alleles per locus. It allows any number of alleles per locus. But it doesn't allow for epistasis. It doesn't allow for gene-environment covariance, gene-environment interaction of any sort. But given this scheme, you can generate phenotypes for different genotypes. So here's a genotype, big A, heterozygote at the B locus, and so on. Here it is pictured down here. And we can go along and we can take the E locus and say, well, what contribution does it make? Well, it makes contribution 0.3. And then we can do, let's see, do I have the rest here? Sorry. Yes, I'm having battery problems in the mouse. That's not a, wait. Yeah. Now we take the A locus there, its contribution is -2. So here we're toting up the contributions of the different loci and of the environment. And we go on and we can add on whatever those tables imply and then draw from the environmental effect distribution a value. Here it happens to be 0.9. And the result will be, when we add it all up, in this case it comes out to exactly 10. And we can go along and do this for each of those genotypes. So here's a set of 5 genotypes. And they each generate phenotypes under that scheme. Now, the question will come up, what is the relationship between the values in different relatives? That's where we're headed. So, oh yes, this model, I just want to mention this because it will come back in on the very last day of the course. We're going to talk not only about measurable quantitative traits, but also using these to model discrete traits. And here we have a trait that has only 2 possible values, 0 and 1. And in the mid-1930s, Sewell Wright put forward a model in which he said, well, we use the general quantitative genetic model, but we apply a threshold. So if our threshold happened to be the number 9, then we'll see that 10 is beyond 9 on the scale, so you get state 1. 8.8 is below 9, so it gives you a phenotype of 0. So here are the thresholded phenotypes, and that turns out to be a very useful model for discrete traits as well. And that comes from the 1930s work of Sewell Wright. But to go back to the continuous traits, I do want to show you also, we'll be, in addition to talking about covariation between relatives, I want to show you a little bit about why we don't just model the whole thing in a Mendelian fashion, why we don't just take all the different possible genotypes and try to figure out their phenotypes. Usually that's not so easy. Here I want to show you a single locus trait with 3 possible values and ask what happens to the distribution and to our methods of genetic analysis as we add more environmental variation. Here we might be in a case where there's a little bit of environmental variance around each of these 3 values. In that case, we could do a Mendelian analysis. We could say, let's try to figure out who is a big A, who is a, who's a heterozygote. And by just seeing the phenotypes, we could classify individuals correctly according to their genotypes. But if we add environmental variation in, these are, by the way, I'm using log normal distributions here. If we add a bit more variation that fattens this distribution, it fattens these 2 enough that they merge with each other. And you now have a case where, well, you could do a Mendelian analysis, but you can only partly tell these 2 genotypes apart. If you put a little more environmental variation in, they all merge into a single distribution. At that point, your ability to do Mendelian genetics starts to degrade seriously. You can also see the same thing happening if I take a trait that is controlled by different numbers of loci instead of having different amounts of environmental variation. Here's a single locus trait with a dominant locus and two possible phenotypes, but with some environmental variation. What I've done here is to scale each of these cases so that the fraction of variance contributed by the genes is the same. It's 30% of the phenotypic variance is genetic in this case. Well, with one locus, you can actually sort of see the different phenotypes of the two possible classes of genotypes. And you might think of doing a Mendelian analysis. If you go to two loci, the distributions start merging. Life is going to get very hard. If you go to 5 loci, now you'll see that although there are discrete underlying genotypes and those express themselves in little distortions of the distribution so small that it's going to be essentially unusable, it's going to be essentially impossible to try to figure out how many loci are contributing there. Here you go to 10 loci, and now you can see the distribution looks very normal. So you're actually approaching a normal distribution, although this isn't technically a normal distribution. It's a mixture of normal distributions, but so many of them that basically it looks like a single normal distribution. So you can see yourself going from Mendelian analysis to being forced to fall back on quantitative genetics, either because there's more and more environmental variation or more loci contributing to a trait. You can also think about interactions between genes. Here's a table of interactions. The genes are not contributing additively, and here we are adding environmental variations, and once again you go from the Mendelian realm to the quantitative genetics realm as we do that. Okay, so let me start talking for a bit about the way that in his quantitative genetic theory, R.A. Fisher partitioned the variance and worked out what the covariances between traits were. And let's imagine a locus that has two alleles, it has 3 possible genotypes. We simply have a scale here counting the number of A1 alleles. We have A1 and A2. So this would be the homozygote A1, this is the heterozygote, and this is the other homozygote. Here's a genotypic effect scale. Suppose the contribution to that model, that Fisherian model, is given by the spots here, so that the A2 genotype contributes this much on this vertical scale, heterozygote and the other homozygote this much. The trait is almost completely dominant, but not quite. The heterozygote is a little bit below the A1. Well, what R.A. Fisher did was a rather strange thing. He did a regression, and he broke the contributions of the different genotypes to the phenotype. He broke them into parts. Excuse me. He broke them into parts in a rather arbitrary way, and you can look at it and say, well, it has no reality. Why is he doing this? And the answer is because it's going to give him lovely expressions for the covariance between relatives. And what he did was to fit, you imagine that this is a population. You also, by the way, are assuming in the Fisherian theory that there's random mating, so you're in Hardy-Weinberg proportions, and also that there is no linkage disequilibrium between different alleles, between different loci. That all the loci are at linkage equilibrium with each other. There are no non-random associations at loci. So this is one locus. What he did is say, let us imagine that we somehow knew these numbers. Let us imagine that we know the gene frequencies of these alleles in the population. In reality, we don't know either of those. And let's fit a straight line by least squares, weighting the departure from the lines by the frequency of that genotype. So if the allele frequency of A1 was P, this delta 1,1 departure here is weighted by P squared. So in doing the least squares fit, what you're trying to minimize is P squared times the square of this deviation plus 2PQ times the square of that deviation, the deviation from the line up to the actual contribution of that genotype, and Q squared times delta 2 squared. Again, you may look at it and say, this is all an exercise in fiction. We don't know any of these quantities. But he says, let us imagine that we somehow knew this and we could do a least squares fit. And in doing a least squares fit, we could then assign to these genotypes, to these alleles, additive effects, which we're here calling alpha 1 and alpha 2, effects which predict the value of the phenotype, the contribution of this locus to the phenotype, and predict it in such a way from these alphas that it will fall exactly on the straight line. And then let's, in addition, imagine that we're measuring the departure, the dominance deviation, delta 1,1, delta 1,2, and delta 2,2, which is the amount you'd have to add to get to the actual genotypic effect of that genotype. So he's breaking up the variation in the contribution of this locus into two parts, one of which are the additive effects and one of which is the dominance deviation. The additive effects are per allele, and they predict the points on the straight line, and the dominance deviation is the departure. Okay, so now that you imagine yourself to have done that, and you basically, the slope of the straight line is the weighted least squares slope that you would get out of that consideration. Okay, so I said this already, we're assuming Hardy-Weinberg proportions and linkage equilibrium. You're assuming environmental effects are independent of the genotype. You're assuming, this is a very strong assumption, that environmental effects are independent in different individuals, which is not true in real life. Individuals live in environments, particularly in natural populations, where you have no control over where they live, and two individuals who are related might also be living in places where they got similar environmental effects on their phenotype. But we're assuming that away. No interaction between loci. Okay. The result of all of these assumptions is to say that under this simple-minded model, all the parts that we add up to get the phenotype are uncorrelated with each other. Technically, they're not independent, but they are, some of them are independent of each other, but all of them are uncorrelated with each other. The two alphas in a locus, the additive contributions from the two alleles at the genotype, the two alleles present in a diploid genotype, are uncorrelated simply because by Hardy-Weinberg they're sampled independently from the population, from the gene pool. The alphas and the deltas in a locus are also uncorrelated with each other. That's by the principle that in a least squares fit, the prediction and the departure from the prediction are uncorrelated with each other, and that's a particular feature of least squares fits. And I'm just saying, trust me that that's true. That's why Fisher ended up defining the alphas and deltas in the particular way he did. Now, by linkage equilibrium, effects at different loci are independent, because genotypes at different loci are independent under that assumption. So those are independent from each other, and the environmental effect is independent. So the result is we have, for a particular genotype, the phenotype predicted as a sum, first of all, of a constant starting point which doesn't vary and is thus uncorrelated with everything, additive effects at each locus, and a dominance deviation at each locus, and these are added up over loci, and finally the environment. All of those are uncorrelated with each other. The whole machinery has been set up and the regression chosen so as to make that true. Well, when you do that, you could rearrange that sum very easily. You could put all the additive effects together and all the dominance deviations together. Here I'm leaving out their subscripts just because I'm too tired to type them, and the environmental effect as well. And by grouping those terms, you can say, well, it's therefore true that the phenotype is equal to a constant starting point plus the sum of all the additive effects at the different loci, the sum of all the dominance deviations, and the environmental effect. So we've broken the phenotype conceptually into three different parts, A, D, and E. And we've made it so those are uncorrelated with each other, and also so the pieces of which each is composed are uncorrelated with each other. Now, if you take the variance of the trait, you can then say, well, the variance of the trait is the variance of this sum. These three pieces are uncorrelated, and therefore it's true that it turns out to be true that the variance of the phenotype is the variance of the A part plus the variance of the D part plus the variance of the E part, and there are no covariance terms because everything is uncorrelated. So Fisher gets this variance component breakdown, but the interesting question is, what can he do with it for covariation among traits? I'm going to assume that you have some familiarity with what a covariance is. If you don't, I suggest you rush out and familiarize yourself with the concept of covariance. It's closely related to correlation. And one of the properties of covariances is that if you take the covariance between two sums, and those sums have a common part, so let's call this the first sum x plus y, the second x plus z. This has nothing to do with, by the way, with sex chromosomes. These are just random variables. It will be true if x, y, and z are independent of each other and thus uncorrelated, that the common part, there's a common part here of these two sums, x, it's in both of them. The covariance of this sum with that sum is equal to the variance of the common part. Now, if we make use of that to compute the covariances of relatives, we can think of two relatives. We'll call their values p sub x and p sub y, their phenotypes, p sub x and p sub y. Under the model that we've got, each of them is a sum of these parts, the additive parts, the dominance parts, and the environmental part. And that'll be true here. And to be more accurate, you'd have to put lots of subscripts in. I've left them out. Subscripts and superscripts, just to keep track of which locus each one is for and which genotype each one is for. But a property of two individuals being relatives at a genetic level is that some of the loci in them are identical by descent. So if you are my half-sib, for example, then for a given copy at a given locus, there is a 1/4 chance that it will also show up identical by descent in you. So for example, if you're my half-sib, and I pick one of my two loci, let's say that you are my half-sib on my mother's side, one of those loci comes from my father and is thus not going to show up in the half-sib. The other one comes from my mother, and she has half a chance of passing it to my half-sib. So there's a half, the probability that I choose the maternal effect, times a half the probability that she passes it on. And so the fraction of copies of the gene in me that also show up in my relative is some number f1, which depends upon the degree of relationship. For half-sibs, it would be 1/4. Similarly, there is a probability of identity by descent where that both of the copies in me will show up identical by descent in my sib. If it were a half-sib, that's 0, because They can't both be identical by descent because one came from my father who's unrelated to the half sib. But if the person was my full sib, then there's a 1/2 chance for each copy that it shows up identical by descent in the full sib. That's independent for the two copies. And they just depend upon Mendelian segregation in the father and Mendelian segregation in the mother, and those are independent of each other. And so for a full sib, F2 would be 1/4. It's the probability that both copies in one individual show up identical by descent in the relative. And we are assuming under our model that the environmental effects are independent and thus and uncorrelated in different individuals. Well, that means that my phenotype is a sum of pieces. The relatives phenotype is a sum of pieces. A fraction F1 of the additive pieces are the same. A fraction F2 of the dominance deviations are the same. And the result is that this is Fisher's, one of Fisher's basic formulas. He didn't write it in this way, but it's this one. The covariance of the phenotypes of two relatives is simply a fraction F1 of the additive genetic variance plus a fraction F2 of the dominance deviation. That comes out of the covariance formulas where the covariance between two sums is the variance of their shared part. And in this case, the sum of the variances of their shared parts. So that's a fair amount of hand waving that I've done, but I'm trying to persuade you that this is at least plausible, that this formula, if the environmental effects are independent from individual to individual, that this formula will hold for covariances of relatives. And now that is very interesting, because what R.A. Fisher has done is to start out with a model that assigns all these alphas and deltas by doing things that you can't actually do in real life. You're looking at a trait. You're looking at the length of my forefinger. We don't know how many loci are involved. We don't know what their gene frequencies are. We don't know what the effects of the different genotypes are. So this has all been an exercise in fiction. But we ended up with a formula that expresses the covariance in terms of variance components, VA and VD. And that means we can go out and measure covariances between relatives. You may not know what loci are contributing to the length of my forefinger, but you can go out and collect me and my full sib. You can go out and collect, let's say each of you had a full sib. I could collect a whole set of pairs of individuals with their full sibs. I could measure their covariances and putting in the values that we know from Mendelian genetics, which for full sibs would be 1/2 here and 1/4 there, we can say, aha, the measurable covariance between the phenotypes of this degree of relative, this kind of relative, should be equal to 1/2 this mysterious quantity VA plus 1/4 the mysterious quantity VD. By doing that for a number of different kinds of relatives, let's say parent-offspring pairs, full sib pairs, half sib pairs, those are some of the most common ones, but you can do more remote degrees of relationship too. All of them will be predicted by these two unknown quantities, VA and VD, with known values from Mendelian genetics for F1 and F2. And so what you've done is to take, by making up all these imaginary quantities, alpha and delta, and then doing the algebra, R.A. Fisher ends up making a relationship that allows us to estimate these aggregate quantities, VA and VD, from observable covariances between individuals of known degree of relationship, of known relationship. So in the end, he gets observables out without actually having to know what all these alphas and deltas are, provided he can make all the assumptions about additivity, about Hardy-Weinberg linkage equilibrium, and independent and independent environmental contributions. So that's actually a tremendous achievement of the 1918 paper. Now, if you look at it, you'll say, you know, this looks a little familiar. It looks a little bit like analysis of variance. It used to be when I was a student, we were all taught analysis of variance. I think that's much less true today. But if you've had exposure to analysis of variance, you'd say, you know, this kind of looks like random effects, fixed and random effects in the analysis of variance. Isn't it a remarkable coincidence that R.A. Fisher's quantitative genetics theory has this flavor of analysis of variance? And the answer is no, it's not a coincidence at all. It's not a coincidence because who was it invented ANOVA? The answer was R.A. Fisher. When did he invent ANOVA? In the years, he first published on it in the years immediately following his quantitative genetics paper. So the machinery was already there, and he was simply making, in the quantitative genetic work, an application of the same machinery that he published in 1922, 4 years later, as the analysis of variance. Each additive effect here is in effect it's something like row and column effects, except you have many levels of this, not just rows and columns. The dominance deviations are two-way interaction effects. You forced a lot of the other interaction effects to zero, and the least squares regression is, by the way, the same machinery that you use to assign effects in the analysis of variance. So it's no coincidence at all. It's all part of the same great development. Okay. And I've already said this, but in effect, you know from the kind of relationship what F1 and F2 are. You get out the covariance formula. So, for example, the covariance between parent and offspring will have an F1 of 1/2 and an F2 of 0. If I have a mother, a father, and an offspring, and I look at the covariance between mother and offspring, that will involve the chance that both copies in the mother show up identical by descent in the offspring is 0. The chance that one of them shows up is 1/2. So you get 1/2 and 0, and you predict that the covariance of parents and offspring will be 1/2 the additive genetic variance. Okay. A word about heritability. Having gone through all this machining, imagining yourself going out and measuring lots of relatives and coming up with values for the additive, the additive dominance and environmental contributions to the variance of the phenotype, we can say what fraction of all of this, of the total phenotypic variance, comes from the additive genetic part. That's called the heritability. It was first defined by Sewell Wright. It was in one of his path coefficient models. He had a path coefficient called little h. This quantity is the square of that. So you have the funny case that the name that we give, the variable that we give for heritability is little h squared. No one ever uses little h itself, the square root of that. The heritability is usually called little h squared. Notice it's not the fraction of all variance that's genetic, because the VD, the dominance deviation, the variance from the dominance deviations, is also part of the genetic variation. So it's not the percentage, it's not the fraction that's genetic. Notice also that we assigned all the alphas, or we imagined ourselves assigning all the alphas, by a linear regression in which things were weighted by the genotype frequencies under Hardy-Weinberg. Therefore, these quantities depend on the gene frequencies. If you take a population and you change the gene frequencies, you will change these additive and dominance variances, and thus change the heritability. As gene frequencies change by things like genetic drift as well, the heritability and the variance components can change from generation to generation. Also, natural or artificial selection can change them, which is as it ought to be, because you might think that if you take a population and you drive all the gene frequencies to 0 or 1 so that there's no genetic variation left in that population, then these terms ought to change. You should not still have additive genetic variance and dominance variance, and in fact the formulas do that. So that's the heritability. You will see it popping up again and again. The fraction of variance which is additive pops up again and again in regressions of offspring on parents. Steve will be making quite a lot of use of it. Okay, here is a simulated parent-offspring regression. The expected slope will be the of a least squares fit. These are parent-offspring pairs. Each point here is a parent value and an offspring value. The slope of the line will be the covariance of the two variables divided by the variance of the so-called independent variable, the horizontal variable here. That's covariance of offspring parent over variance of P. That's going to be 1/2 of VA over the VA plus VD plus VE. In other words, it's a half the heritability. That's what this slope ought to be. If we then take in a regression like this and say, let us imagine ourselves choosing offspring by taking the parent, let's say in this case it's the father, and considering only offspring who are the offspring of fathers whose value was above this red threshold. So here's a value a little bit less than 12. We decided to only consider offspring above that point. First of all, let's only consider offspring who are right at that point. If you consider the offspring of individuals whose phenotype, whose father has phenotype 11.8, well, you go up here and you say, there they are, there's a thin slice through here. There, in that live all the offspring who are offspring of a father of phenotype 11.8. What is their average? Well, under the case where everything is normally distributed, that will be precisely on the regression line. So the average of offspring of fathers whose values are here is gotten by just going up here, bouncing off the regression line, and there is the average of offspring of parents who are at that value. Furthermore, if you take all the individuals who are above a cutoff, then for each value of fathers, there is an average value of the offspring. Here are their offspring, and what I have to persuade you of is the fact that if I take all the different fathers, for each one I take what the average offspring is, and I weight them according to the frequencies of those fathers, what I'll end up predicting is that if this is the mean of the individual of the fathers that we save for breeding, here's the cutoff, might be at 11.2 or something like that. There are many, there's a distribution, it's a tail of a normal distribution of fathers. The average of their offspring will be gotten by taking the average father that's preserved and bouncing off the line. That's not obvious, not entirely obvious, but it comes out of the linearity of everything that I've been doing here. So you can use, if you knew the heritability, you can make a prediction, in this case, that from parents who are a certain distance above the original mean, we can predict what the distance of the offspring will be. The result is what's been called the breeder's equation. And that is that if you breed from parents who are, in this case, by truncation selection, if you breed from a collection of parents who are a known distance above the original population mean from which they were, of the population from which they were drawn, that difference is called the selection differential. So if we have a population whose mean is 10 and we choose males whose mean is, whose mean turns out to be 11.4, and females whose mean turns out to be 11.4, and we breed from them, it will turn out that the resulting change in the population mean of the offspring will be equal to the heritability times that selection differential. And you'll be seeing this breeder's equation again and again as Steve talks to you in the next few days. So if you're doing selection not just on the males, but on both males and females, the response is equal to the heritability times the selection differential. And this is the basic breeder's equation. Okay, I'll finish up by just giving you some references here. The projection PDF is posted at the website, so you can go there and get the references. Here's R.A. Fisher's 1918 paper, and I note that it's an impossible read. Anyone who claims to have sat down, read it, and casually understood it, I'm not going to believe you. Here's Jay Lush's book. It was published in many editions called Animal Breeding Plans, and it was very influential in spreading the word among animal and plant breeders. Its 1943 edition is readable at this link. By the way, the links in this projection should be live links. When you get the PDF up in something like Adobe Acrobat Reader, you should be able to just click on them and it will go there. Two great texts of quantitative genetics are available. One is Douglas Falconer's 1960 book, An Introduction to Quantitative Genetics, which was an absolute classic at the time. It's the book from which I learned theoretical population genetics. I had the great pleasure of knowing Douglas Falconer when I did my postdoc in Edinburgh 8 years later, and I was able to tell him I had learned everything that I knew in the field from that. Trudy McKay, or Trudy McKay, depending on who you are, how you pronounce her last name, who's at North Carolina State University, became an additional author in later editions of Introduction to Quantitative Genetics. I think its most recent edition is the 4th edition. It's a classic text, and so if you want to read up on quantitative genetics, it's a great place to start. So is the book by Lynch, much bigger book by Lynch and Walsh, Michael Lynch and Bruce Walsh, published in 1998, Genetics and Analysis of Quantitative Traits. They have a second volume, which is just about to come out. Chapter drafts of it are available at Bruce Walsh's website. The volume which is about to come out is the first volume of volume 2. Have you got that? There's volume 1, which is an actual volume, and then there's volume 2, which will consist of 2 volumes, volume 2 part 1 and volume 2 part 2. Andy Sinauer is going to be publishing in the autumn the first part of volume 2. Another place you can go is I have basically a theoretical population genetics textbook called Theoretical Evolutionary Genetics. It's been available for many years as a free download from my department. It will remain that as I revise it. It's getting to look more and more like a publishable book. I will not be taking it down. Any publication, any arrangement I make for people to buy printed copies will be made in such a way that the online free PDF download will still be there. In Chapter 9 of my Theoretical Population Genetics text, I give you, with some more details, the argument that I basically gave you here. So you can read that for another run through my presentation of this. So that's all for this introduction. I hope it has been of some use. We're supposed to have half an hour of discussion. I think we'll probably not end up needing that much, and that'll enable us to catch up some with our schedule. After that discussion part, we're going to take a break and then go to Steve's first lecture for the last part of the morning. Now, you have sheets of paper you can hold up, you have microphones, but whatever you do, use a microphone. Try to think of questions. This course is structured in such a way, when Steve and I first designed it, we wanted to get away from a situation where we simply bombarded you with lectures and then you had no time to discuss them and were bombarded by more lectures. So we allocated half-hour slots after every lecture for discussion. And really, it's up to you to make that work. And we really do want to hear from you. And if you have a question that you think, well, it's probably a stupid question, it's probably not a stupid question. There probably are a number of other people here who have exactly the same question in mind. So please don't be timid at all about asking questions. So if anyone wants to ask a question, raise your hand, and then somebody who has a microphone will get you a mic. Flip the mic on, wait 1 second or so, and then point it at your mouth, not at the ceiling, and then don't be modest and mumble. Speak out egomaniacally, okay? So you're all familiar with all this stuff, and there are no questions. You don't have any doubts. This is all assimilated in you. Everybody understood everything I said? Okay, maybe somebody didn't. Okay, now you have a mic. She needs a mic.
Thank you. So if you could go back to your slide 17, where you have the first definition of additive effects and dominance deviations.
I'm trying to read the tiny little print as to which is 17.
The first plot where you do the linear regression.
There.
Yeah.
Yeah.
I'm not very clear on what the horizontal line means. Is that the average of additive effects, or where you're measuring the alpha 1 plus alpha 1 from?
Okay. The horizontal line is actually number of copies of A1.
No, but in the y-axis.
Oh, you mean what
The
this dotted
dotted
line is
yeah.
here? It's actually the mean. It's the mean of the. In effect, actually, it's 0. That is 0. So if something had 0, if a genotype made a 0 effect, it would be right on that dotted line. I think that's probably the simplest.
Okay, so the heterozygote has 0. Let's say, would you define it as a 0 additive effect, and then the
No, actually, they're
alpha
in the
A1.
in this example here. There is a small additive effect of the heterozygote.
Ah, I see. Mm-hmm.
I was trying to make sure it wasn't right at 0.
Right,
I'm not
yeah.
sure. You
That
can
wasn't
see
clear.
the purple head of the arrow
Yeah.
there, but
So they come from your first slides, like the values you define from your first slides for the
the
heterozygote.
things that were in those braces are these there are these 3 values here, here, and here. Are what are in the braces.
Okay.
And then when you then you fit the least squares line, and that defines, for example, the difference between the fit, the prediction from the fit, and 0 is going to be 2 of these alpha 1s. This will be 2 of these alpha 2s, they'll be negative. 2 of these alpha 1s, they'll be positive. This is an alpha 1 plus an alpha 2. They don't cancel out perfectly.
Thank you. It's clear now.
He needs a mic.
So I have a question just about if one was to in practice implement a regression method to estimate heritability. So just standard linear regression. One of the assumptions I think is that the X value is estimated without error. So in your case, the parents are estimated without error, right?
Yes, it is. In the machinery I've given you, let me see if I can get us down to, to here. This is a bivariate plot of a bunch of points. We went out and imagined ourselves measuring 200 parent-offspring pairs. We measured with perfect accuracy, but that does not mean that the parent has no environmental variation. It does not mean it has no It has no developmental sources of noise, for example. All it means is that we didn't go in and in addition commit a lot of measurement error.
Yeah,
So
yeah.
we've left that out. If you want to, it in effect is an additional source of environmental variation. If I go out into a population and I just do a lousy job of measuring everybody, well, I've just added a random variable onto this phenotype that I come up with. So here we're just saying, well, whatever the sources of environmental variation, we're not having any measurement error. We're not worrying about that.
Yeah, and I guess I know that there are regression methods to account for measurement error in both the X and Y variables. So I was wondering.
What you're trying to do there is get a regression that doesn't reflect that measurement error, but that removes it from the analysis, in effect. That's a complication that I'm not going to get into, and I don't think we'll get into it at all in the whole course. But it would be an additional level of noise that you would have to worry about if you wanted not to count the measurement error just as environmental error. And you wanted instead to know what fraction of the true phenotype is additive genetic or dominance or environmental, then you would have to do something like that.
Okay.
Have you got, yeah.
So
Pointed at your mouth.
So I have a question about like one of the first slides about what do you, because like when I heard about the contributions of an allele towards a phenotype, I thought about like measuring something, but I also see that there are some negative numbers and so on. So what's actually the contribution, and what's the definition, the concept of the contribution of an allele on a phenotype?
And that you're concerned with the issue of how could an allele have a negative contribution to a phenotype?
I mean, you can see how something can like prevent or inhibit or something like that, and that can have a negative effect. But how is it measurable? Like, how do you actually get to the number?
If you knew all the genotypes of every, if I knew for my length of my forefinger that there were 127 loci, and here is exactly what each genotype contributes. Of course, we don't know that. If you knew that, you could imagine taking one of the loci, and you could imagine looking at the different genotypes, and say, well, on average, their contribution is 0. We'll set the start, if we set the starting point, that mu in the model, to be the population mean, then a given genotype might contribute an amount that is less than others, and so it would tend to push the phenotype below the population mean. In that sense, it would be negative. Only in the sense of comparing that genotype's average phenotype to the average phenotypes of all genotypes at that locus, and it would be below the population mean. So it isn't, there's no, all that's involved is that some genotypes are adding less than others. And we're starting from the population mean, so we have to be negative to get to the contribution from those genotypes. It's not imagining something mysterious happening in the development of the trait that involves very strange values. It's just some things are giving higher values and some things are giving lower values. And we start from the population mean when we think about these things.
So it's basically like how strongly one loci is pushing or pulling the phenotype.
Yeah, and in the model here, they're all just adding in.
Mm-hmm. Okay.
They're either adding in smaller amounts or adding in larger amounts, and in the end, you get a distribution. It has a mean, and so those that added in smaller amounts are book kept here as negative contributions. I have to warn people, by the way, that scratching your ear in this class is very dangerous. I will call on you. Yeah. I thought I saw somebody do that.
I have a question about the F1, F2 value.
Yeah.
I didn't get that. Could you please explain that again? And also, what's the relationship between F1, F2 and the relatedness?
F1 will define as the probability, if you are one of a member of a pair of relatives, like it's a parent-offspring pair. If you're the parent, and we take a copy of a gene at a locus, at any locus, what's the probability that it shows up in your relative as a result of identity by descent? So now, it could be that that same allele also occurs in the relative, not because it's identical by descent, but just because it happened to come in from another ancestor. So it's a conditional probability given a single copy of the gene that it appears in the relative as a result of identity by descent. And it is, let's see, I have to, the coefficient of relationship, you mean the one that's used, for example, in kin selection arguments? I'm trying to remember, is that a factor of 2 off? It is, it is basically the same quantity as used in a kin selection argument for the F1. The F2 does not get used in a kin selection argument. But the F2 is the probability that both copies are there, and you can usually work that out. In the simple cases like full SIBs, it's just the square of F1, but that's not always true.
You know, I'm not even sure there's a name for F2.
Well, it doesn't, you know, in kin selection arguments, we're usually thinking of an allele that's rare in a population, and we're asking what's the probability that it shows up in relatives, and nobody really wants to know this one in that case. And if they do have a name, it's not standard. It's not something that everybody, you know.
It's not something Joe and I ever can remember if it exists.
Yeah. I'm sorry, you're speaking without a microphone. That's a fatal sin in this case, and
in the
I can't hear the microphone. Could you test it and see if it works? Okay, please, yeah. Okay.
There is a term in the Falconer book for F2.
I don't care. I think Falconer made such a term in the hopes that it would be useful, but nobody else has picked it up. He needs a mic. I saw you earlier, and I'll get to you, but he got my attention first. But I'm glad there are more questions.
So it's a follow-up question on the F2. So if the value of F2 for full SIBs is 0, is it to get a non-zero value, do you need inbreeding? Is that?
Wait,
how
the
that's-
value of F2 for full SIBs is 1/4.
Oh, it's 1/4. Okay.
Because you can get, if I have 2 copies in me, one came from my mother and could show up in the full SIB. The other came from my father, but since it's a full SIB, could also show up. And those are independent because they're different Mendelian segregations, and they turn out each to be 1/2, and it comes out to 1/4. But I'm sorry, you were asking a question based on that.
No, that was the
Microphone. Use the microphone.
that was the question.
Okay. Now there were a couple more questions. You had, before we get to the back, you had your hand. It's not when you put your hand up, it's when I saw it. So, yeah.
Yeah, I had a question about your slide on response to truncation.
Yeah.
So I'm not sure I completely understood sort of truncation and why you would do it and what effect that will have overall.
Okay. Truncation selection is very widely practiced by animal and plant breeders for the simple reason that if you're trying to make the fastest possible progress, you'll breed from the highest individuals in your population. So it's a very common kind it's a very common kind of artificial selection out there in the real world. The breeder's equation that I showed you applies to all kinds of schemes of selection, not just truncation selection. So I was here imagining truncation selection and making the point that while for each little slice from this, each tiny little slice from this axis, we can bounce off the line and make a prediction. It then turns out that if I do that for all of the slices and I weight the responses appropriately, I will in effect be taking the mean of these and just bouncing it off the line. That turns out to be true. That was the point I was trying to make here. I chose truncation selection simply because it's one of the most common kinds of selection. But for other schemes where I might, for example, select a bunch of individuals near the population mean and some from here, and maybe not any after that, but maybe a few from there. I could have some sort of crazy scheme like that. It's still, the breeder's equation is still true. If we take the mean of the selected individuals, find that on the curve and bounce off, that will make the correct prediction for that scheme of selection, which is not truncation selection.
Okay, thank you.
Okay, you had a question back there and need a microphone. No, I can't hear it.
Off. So.
Okay,
Yeah.
now it's on.
If we have an inbred, so this relates to the F1, F2 question again.
Yeah.
If we have some level of inbreeding in a population, but we still have random mating in our design, that alleles in per locus are still, I think, uncorrelated if we have the random mating, but the inbreeding will inflate the F1 and F2 coefficients. So how much that messes up our estimation?
It will mess things up, but I question your assumption. If we have random mating, then we don't have inbreeding.
No, but I mean, we have inbreeding in our base population. So our population is already inbred to some extent.
So you mean, for example, it might have undergone genetic drift from its original situation.
Right.
I think in that situation, all of this continues to apply, provided you forget about the inbreeding and just consider the population to be a random mating population. Then all this algebra applies. But the additive genetic variance that you're estimating is the additive genetic variance in the current population, not in the original population relative to which you are inbred.
Mm-hmm.
The original population that was there before the genetic drift. Okay, we've provoked Steve, who's going to correct me.
Yes. Okay, you've provoked me only because I was thinking about this just last night. And what I discovered online, so to your list of assumptions, you could have added no inbreeding. But so the
Well, I
reason,
said random mating. That's good enough.
okay, but so if you go online, you can find notes by Bruce Weir, who goes through the whole scheme that we went through today and relaxes the assumption of no inbreeding. That is to say, pairs of parents that you choose are not inbred. And what happens to the whole formulation is you get the same coefficients F1 and F2 and additional ones for epistasis, but you also get a coefficient of inbreeding. And so it's just added into the scheme, missing in the case of assuming everybody's outbred when you choose parents. So
I will add to that I don't think anybody ever uses that stuff. Now,
I've never used it, but they're there. His notes are there for you.
Yeah, and I have to say, we're live streaming. Bruce is at my university, up in his office on the 21st floor, or 15th floor of the university tower. Hi, Bruce. I'm sorry, but I am saying that I think you'll find it relatively rarely used. Provoked him again.
What Bruce has accomplished.
I'm a great admirer of Bruce. Okay. There were more questions. Please, you have a mic?
I just have one more question about the breeder's equation. for the individuals that survive, say, make it past truncation selection, is the breeder's equation assuming equal fecundity across all of the surviving parents? And
Yes.
it is. And
It
so
is- it is. You'd
if-
have to, if you had parents who differed in fecundity in a way that depended upon where they were on the scale. Not, I mean, of course, at random some individuals have more offspring, some have less, but if that happens unconnected with the phenotypes, it won't change the prediction. But if you knew that the further out one got on the scale, the more sterile everybody was, you'd have to weight in computing the selection differential, you'd have to weight it by their resulting fertilities. Yeah.